Good afternoon, everyone. Today I will present some work for my PhD focusing on the investigation of face processing in emerging ASD. And like my colleagues in this symposium, I will focus on the social and communication domains of autism. And although autism is usually treated as a unitary category, it is rather heterogeneous. And this high level of individual variability is evident both in terms of clinical manifestations and biological underpinnings. And the recent framework suggests that ASD is a syndrome emerging from a critical aggregation of early interacting liabilities that can be best described as endophenotypes, namely quantitative and heritable traits that can be measured continuously in the general population. And face processing represents one of the most promising candidate endophenotypes for ASD development. In fact, infants who go on to develop autism show altered social behavior already in the first year of life and can be seen, for instance, in reduced interest in looking at faces. And these behavioral symptoms, behavioral manifestations are accompanied by neural alterations as indexed by event-related potentials or ERPs. And you can see here an example of a waveform in response to face and visual noise. And I have highlighted three components, the P1, N290, and P400, as they have been previously shown to be altered in emerging ASD and in children with ASD. And you can see here some of the findings. And in particular, P1 is a sensory-related component that can be modulated by selective attention, whereas N290 and P400 are more later, higher-level cortical stages of processing that can be modulated by deep engagement and attention to the stimulus. We must say that previous studies have shown diverse findings supporting the idea of altered face processing in emerging ASD. However, these diversity can be due to consistent differences in lots of different specific aspects of face processing, but it can also be due to individually specific profiles of specific alterations or different profiles of alterations coherent in specific subgroups of ASD. So how can we test whether one or another of these hypotheses are true? Well, group-level studies have not succeeded in doing so far, so we opted for novel approaches, individual-level approaches that could help us understand the pattern behind such diverse findings in altered face processing for ASD in infancy at least. And what we found, well, what we chose to do was to take a perspective approach. And like Dr. Siddiqui said in her presentation earlier today, that's a very variable design based on infants considered to be at elevated likelihood for developing autism based on having a older biological sibling with ASD already. And it allows us to investigate the causal mechanisms in terms of understanding the emergence of ASD symptoms. And in this case, for our study, we collected ERPs at 8 months and used those to predict categorical outcome at 36 months, the ASD diagnosis. And to do so, we used pattern recognition, in particular the support vector machine classifiers. And basically, we have a set of features or just measures per subject, and each point is just an infant here in this plot. Red is for ASD group and green is non-ASD group, and the algorithm finds a way to separate the two groups. It just finds a plane to separate between ASD and non-ASD infants, and then you feed the algorithm with new individuals and it just decides whether it's on one side or the other of the plane. So doing so, it just predicts individual outcome. And the next step in our study was to try to understand the heterogeneity of face processing in autism by stratifying the sample into subgroups, and we used Bayesian clustering for that. Let's first describe the measures that we used. The infant was sitting on the parent's lap looking at the screen, and we showed them faces with direct gaze, averted gaze, and visual noise. That's just a scrambled face in here. And then we had different contrasts, the gaze shift contrast, comparing gaze shifts directed towards versus away from the infant, static gaze contrast, comparing faces, static faces with direct or averted gaze, and then the face noise contrast, comparing faces with just average eye gaze direction to the visual noise. Then we obtained the waveform per condition, and we did that for 145 infants at elevated likelihood for developing ASD. And our first aim was to predict clinical outcome at an individual level, and to do that we used the classifiers that I showed you before, and as measures we used all the different amplitude and latency for different components in response to the different stimuli, but also to the contrasts. And we used all of them together, but also subgroups. So we selected the features to see whether there were different groups of features that were most predictive. And for selection, we used two different approaches, top-down and bottom-up selection. For the top-down, we just based on the task, the different contrasts, whereas for the bottom-up, we used the genetic algorithm. And basically, that's inspired by the process of Darwinian evolution. So you start from an initial population of feature sets, a group of feature sets, and then you compute the fitness, and in this case, that's a classification accuracy indexed by the area under the curve, and then the evolution starts. So you have the next generation of feature sets composed through crossover, mating, and mutation of the original feature sets. And then you go on and on and on again until you reach the end of the evolutionary process and you get the optimal feature set here. But we also wanted to know whether there were features that were most relevant for prediction, and to index such sort of relevance, we used the so-called highest incidence set based on the incidence of each feature in the top performing classifiers throughout the evolutionary process. So we just selected the top performing classifiers, looked at which features were used to predict, and then looked whether different features were selected repeatedly in these top performing classifiers. Then we compared all the different groups of features, all the different classifiers through shuffle tests. And here you can see only the findings for those classifiers that performed significantly different from chance level prediction. And the best performing one was the optimal feature set. You can see all the different features in here, but it's enough for you to know that it's just a diffuse pattern of alterations in response to faces and visual noise. And this could predict individual ASD outcome at 3 years with good classification accuracy, good sensitivity, high specificity, and even good positive, negative predictive value. And now we wanted to know whether this diffuse pattern was due to all the infants developing ASD that show diffuse pattern of alterations of face processing, or different infants show different alterations. So what we did was the stratification analysis through clustering. We used the same measures that I showed you before, but just not the contrast, just only the single measures. And I only use the ASD group. And then the Bayesian clustering consists, you have here the individuals, and you test through Bayesian tests whether it's meaningful to group pairs of individuals until you just reach a level of subgrouping that could be the entire cohort if there is no subgroup, or just meaningful subgroups. And we found two, one with 13 and the other with 19 infants. Then we compared the two groups in terms of face processing to see the characteristics that made them be separate groups. And here you can see that the red cluster, cluster 1, is characterized by sort of increased selective attention to faces with averted gaze and faces in general, as indexed by the P1 amplitude, and increased engagement to the same stimuli as indexed by the N290 amplitude. And this was accompanied by increased symptom severity, although significance was not retained after correcting for multiple comparisons. But we also compared the clusters to the non-ASD group to see which were the differences between ASD subgroups and non-ASD group. And we found that the cluster 1, the red cluster, was the one with more specialized alterations in later stages of phase processing, in particular P400 amplitude in response to static phases, whereas the blue cluster was showed more of a diffuse pattern of alterations in processing of phases compared to the non-ASD group. All of them had higher levels of symptoms, but only the red one, the red group, showed the reduced motor communication and daily living functioning compared to the non-ASD group. So the red group seems to be the most severe one and has a more specialized alteration in response to phases. So the take-home message is that our findings support the hypothesis that diffuse and individually heterogeneous alterations in social processing of phases contribute to the emergence of ASD overdevelopment, and we hypothesize that it does so as failure to develop expertise in processing of phases has downstream negative effects, eventually resulting in a social symptoms characteristic of ASD. So we support the idea that ASD is a syndrome emerging from diffuse and interacting alterations early in development. And although prodromal intervention is supported at the level of the group currently, individual prediction of ASD in the first year of life might be crucial to enable early targeted intervention and to identify those infants that might benefit most from that intervention early in development. And we also showed preliminary support for subgroups, coherent subgroups of phase processing in ASD. And these subgroups might be more relevant target groups for intervention, might improve predictive power, but they might also improve our understanding of specific physiological mechanisms for the emergence of ASD symptoms. In terms of limitations, we of course need to integrate different kind of data like MRI, tracking, or even genetics to improve prediction, but also to refine our understandings of these possible subgroups in ASD phenotype. And it would be useful to see whether the ERP parameters are sensitive to intervention effects to use these ERPs and classifiers as proxy measures for intervention. And finally, we need to consider methodological limitations of machine learning. So we had a relatively small sample size, and although we use whole data validation to test reliability of our model, it is important to test the generalizability in an independent sample, especially considering feature selection. And I want to thank you for your attention. Thank the supervisors, my colleagues and families that permitted this all. And if you have any questions, please write them down in the chat box. I will be happy to answer to those questions live on July the 8th. And otherwise, you just send me an email. Thank you.